Well, good evening, everyone. For those of you who don't know me, I'm Trevor McMillan. I have the great privilege of being the Vice-Chancellor here at Keele. And part of that privilege is to chair a number of our inaugural lectures, a series which we take very seriously here at Keele. We try to advertise, we ensure that the series continues at a time when many other universities are actually not doing inaugural lectures, which I always think is a great shame. It's a time for the university to really show off some of its strengths, in particular in research, a time for us to show those strengths to the outside world, and indeed a time for some of our new professors to show their mettle in front of some of their colleagues, and actually show themselves as good examples for our colleagues. Because all of our academic colleagues at Keele do have that ambition of being a professor one day, or at least the majority do, and actually to show how good you've got to be in order to get that accolade, I think is an important thing for universities to do. It's also a pleasure to come to lectures like this to see a broad audience. And this evening, I understand in particular, for example, a broad audience from around the university, from two faculties. It's certainly a pleasure for me to see the word biological up there as a biologist by background, but also the audience from outside the university and indeed friends and relatives. And it's not often that I can stand up and say at an inaugural lecture that it's a particular pleasure to welcome people from Australia this evening to the lecture. So this evening's lecturer is Professor Melissa Mather. Melissa was born and educated in Brisbane. She obtained a Bachelor of Applied Sciences degree with first-class honours majoring in physics and minoring in maths, and that was obtained from the Queensland University of Technology. She stayed at the QUT to carry out her PhD in the Centre for Medical and Health Physics. On completion of her PhD, she moved to the UK to take a research position at the University of Nottingham, where she stayed for 12 years. And during this time, she worked on the development of sensing and monitoring techniques for the use in regenerative medicine. And she concurrently held a strategic research fellowship in the biomaterials group at the National Physical Laboratory. In August 2015, Melissa moved to Keele University to take up her current post, where she's establishing a research group in biomedical imaging. And just to pick up on one achievement in a CV, which is hugely impressive. Most recently, she was awarded a European Research Council Consolidator Research Grant, and that fellowship provides 5 years of funding to develop new techniques to study transmembrane proteins in their natural state. That in itself is a major, major achievement. To get that amount of prestigious funding actually will mean that the research is not only respected right around the world, but also gives an even greater chance to make a greater impact with the research itself. So it therefore gives me great pleasure to welcome Melissa Mather, Professor of Biomedical Imaging, and invite her to present her inaugural lecture, Using Pictures to Explore the Biological World. Melissa.
Great, thank you, Cheryl, for the kind introduction, and thank you to the audience for taking the time to come and listen to this lecture. I hope you'll find it's a good investment of your time. So today, or this evening rather, we're going to talk about using pictures to explore the biological world. And in particular, what my career has been looking at is innovative technologies that have assisted us in our biological imaging. And what I want to do to start with is to take a historical perspective. So biological imaging has been a big topic for many, many years. And when I say many years, if we consider illustration here from the 13th century, we can see without too many innovative technologies, we've got illustrations showing the veins. The innovations at this time more related to the pigments and the sorts of binding materials that are related to producing the parchment itself. we move forward to the 14th century, people were starting to get very interested about the anatomy of the brain, quite a fascinating structure. But you might question yourself and say, in the 14th century, what types of innovative technologies did they have to look at the brain? Well, it turns out it's a hammer and a chisel. I'm hoping this person wasn't actually awake, but it shows that this approach, although it's depicted in quite a crude way here, that is dissection, really carried on through history as being one of the main modalities that we can get good detail of the biological system. So rather than x-rays and sort of other non-invasive techniques where they're looking more at the macro scale, it's through these sort of approaches of dissection where we really look at the biology. So now one of the seminal works was carried out in, this is a work here published in the 16th century. And this text, in fact, was used for many, many years and formed the basis of the understanding of human anatomy and in medical teaching. And not only was the scientist here a tremendous illustrator, he also happened to live near a prison where executions were carried out. So he had plenty of samples for his investigations of the body. So that is one coincidence there. But again, just reiterating that the innovation here really was coming from these surgical techniques. Moving on in time, people wanted to not only look at the structure, but they wanted to look at the function and see how do these organs work, what parts of the organs and how they're related to each other. And at this time, a problem of looking at the brain particularly was that after death, the brain pretty much becomes a semi-fluid. So trying to look at the physical structure is quite difficult. But at this time, the innovations were related to fixatives. So some chemical strategies were produced so they could study the intact brain. So what we've seen here is we've seen some dissection being the main technology here, and I can't imagine what the smell may have been in this picture when it was taken, hence perhaps their expressions on their faces. But it goes to show that this is up into the 19th century, this is playing a crucial role. So the surgical techniques really are giving us access to the biological world. But when we want to look at things on a cellular level, and we want to look at things, in fact, on a microscopic level, we, as you could imagine, need a microscope. And Robert Hooke famously investigated biological systems. And you can see a picture here of Hooke's microscope. It's very basic, using a candlelight. But this concept of illumination of samples and using combinations of lenses is really carried forward to the modern day. And microscopy, I guess that's the message, the theme of this lecture plays a tremendous role in understanding of biological systems. So Hooke published this book here, which became really represented the first book that was published by a scientist, including images of biological systems under the microscope. And it was this picture here that you can see of cork, where Hooke coined the word cell for the first time relating to a biological object. He said that it was reminiscent of the monasteries and the cells of the monks, and hence he came up with this term. So now we move into the 19th century, and we're getting some tremendous pictures of the structure of the brain. And we can see here Cahill, who was a neuroscientist, and I think it's always is an important message in this self-portrait here, is to do a job that stimulates you, as you can see. He's maybe had a hard day, I don't know, but regardless, he's produced some tremendous tremendous descriptions of the structure of the brain, and in fact was awarded the Nobel Prize in Medicine. So this is this historical perspective, which just shows that from a long, long time ago, there was great interest in biological imaging, and that interest extends today. But I want to shift gear and take you on a bit of a personal journey as to how I started to have an interest in science. And as it turns out, I'm looking through some documents when I was home recently. It turns out I had a keen interest in science from an early age. So here I am on the first day of school. Notice there's no scale bar, so I'm not sure if that is an A4 book. I'm hoping it isn't, else I'm a midget. And you can see I'm prepared there with an enormous school case. All I can think is that laptops were a lot bigger in the 1980s. But so here I am raring to go, and looking through some school reports, I found this, and I'd like you to at least focus on the science section, which says there is a keen interest. And to my dismay, the physical education I personally find quite embarrassing. Music experiences some difficulty. I think that's pretty harsh for a 6-year-old, isn't it? I don't think I could get away with that these days. But I have carried on participating in sport, since then. And so for that reason, I just need to set the record straight that it did improve. Here I am. I'm in the school softball team, so that must have included a coordination. And I get to senior school, and I'm looking very excited on the end there. Captain of the tennis team. So I just wanted to set the record straight there. Coordination did improve. However, it turned out the keen interest in science also continued, and here I am in the equivalent of my A-levels. And I was fortunate enough to be selected to go to a national science summer school, so that's where all the cool kids go on their summer holidays. It's a 2-week science school. And here I am in one of the physics labs at the Australian National University. And it was not long after I sat in that chair that I decided I wanted to be a physicist. So I have to become a physicist. So I went to university, and as Trevor said, I took a science degree majoring in physics with a mathematics minor, and then, of course, embark on the journey of further education. And becoming a physicist was something quite special to me, and I think it's summed up very well in this book, which was written by a chap who spent some time with Richard Feynman. Richard Feynman being really one of the most remarkable physicists of the modern time. And a particular part of it that says to be a physicist is to have enormous potential to change the world. Well, that's pretty something. And it's also to share a proud history and tradition. So I think it's a real privilege, and certainly something that I wanted to be able to do, and hope that by the end of my career I've at least lived up to that sort of potential. So what does a physicist do their PhD on? Cancer, of course, because that's an obvious thing for a physicist to work on. So I wanted to pick a topic which had some meaning and some motivation and some impact. And it's the harsh reality that every year 14 million people worldwide will hear the words, "You have cancer." And that was a sentence that my poor father heard just before Christmas. So it's something that affects us all, and so it's certainly a motivation for us becoming researchers. So what has this got to do with physics? Well, about 40% of people who have cancer have radiotherapy. Okay, so radiotherapy is using radiation to kill off the cancer cells. And medical physicists get involved with this. And the first stage is they will, you generally have an image, so you have a medical image to find out where the cancer is. And then the physicists will do some simulations and work out where they should irradiate you and how much. So if we have a look here, so what they do these days is they radiate you from multiple directions, so they can get the maximum dose in the area where the tumour is. But to make sure they get the dose in the right place, they have to do something called dosimetry. So radiation dosimetry is all about making sure these advanced linear accelerators that deliver the radiotherapy give it to you in the correct dose and in the correct location. And you can see here, this is a chap from the National Physical Laboratory, and they've got a big tank of water, and they've got a little detector that they're scanning around. And that is really the gold standard of what they do. But my PhD was based on a different type of dosimeter, and that was a polymer gel dosimeter. And what made that different is it's three-dimensional. So how that works is we take a mold. So say, for example, the person is going to has a tumor in their head. We fill it with a jelly, take it to the hospital and irradiate it. And where the radiation has gone, we get some cross-linking of the polymer. The amount of cross-linking of the polymer relates to the dose. And subsequently, we can take this to the MRI machine and we can read out the dose. And then we can compare that to this mathematical model of the radiation dose that was planned and see how well it verifies. But, I mean, this technique works. It's great. It's fine. But MRI scanners are in scarce supply, and not everywhere has them. So the topic of my PhD was, can we find a way to do this with ultrasound? Because it's smaller, it's cheaper, and it's more accessible. And it turns out we can. So what we've got over here is we've got some acoustic properties. So it's relating to how fast sound travels through that material. And it turns out that the speed of sound changes with the amount of radiation we give. And this insert curve is a sort of result you get from MRI. And what's interesting is that we reach a dose with the MRI scanner that there's not much change in the result. Whereas when we look at, this is how much the sound is attenuated by the gel, we can see that this change occurs over this whole range. So it means that if we use these ultrasound parameters, then we can actually assess a greater range of doses. So that's very nice, but here you can see the high technology we had at the lab. There is this rudimentary ultrasound imaging system, and took this phantom here to the hospital and irradiated it with a square beam of radiation, and was able to reconstruct the area of where it was irradiated, just so to prove the proof of concept. And what's nice is I did a recent literature search and found that, in fact, even recent papers are still discussing this work, so it's very nice that the work that was initiated in my PhD is carrying on. During my PhD, I had the privilege to work with Tony Collins, who worked at the Commonwealth Scientific Research Organisation. Sadly, Tony died a few years ago, but he was a tremendous physicist and a tremendous person, and it was a real, as I said, privilege to work with him. Francis Duck is based in Bath, and he's really one of the world authorities in medical ultrasound, and again, it was a privilege to be able to work with him during my PhD. So I still had some interest in creating interest in science. So here's me on a train station platform. So during my PhD, the university got a train and kitted out all the carriages with science exhibits, and we went to outback Queensland. So this is in Longreach, and if you've ever looked at a Qantas aeroplane, it's got Longreach written on it, because that's sort of where it all started. And the fun thing about that was I got to drive the train. You always need a plan B. Not so good for the kangaroo who didn't get off the track, though, despite tooting the horn. Right, so I'd finished my PhD, and really our head of department sort of kicked us all in the pants and said, if you want to become a sort of better rounded scientist, you need to go abroad and see what other opportunities there are. And so to quote Disney's Aladdin, I entered a whole new world. Now, being from Australia, I would have thought England pretty similar. There are some subtle differences, however. road sign in Australia, road sign in England. Now you've got to pay attention to this one. Power socket, pretty simple. Upside down. You're upside down, by the way, not us. A radiator. Now this is the only place I'd ever known a radiator exist. You've got it in the house. Never ever seen that before. And as Jeanette will know, when I was looking to rent a place and it said gas central heating, well, to me that meant there must be gas in there. Seemed logical. Anyway, so I'm in this whole new world. And so we have a bit of a shift from looking at cancer. And I had the privilege of working with Professor Richard Chalice, who's here in the audience. And Richard's work was really on in-process monitoring. In particular, this applied to a lot of industrial materials, so talking about high volume and high value chemicals, looking in the food industry and so forth. And Richard really pioneered work where we've got, these are some ultrasound transducers here on a pipe. So this is in process in a if you like, a plant that's making these materials. And so I had a real change in gear from looking at biological things to these really exciting, sorry Richard, slurries. But, however, I did learn a lot, and I still have a bone to pick with Richard because he made me solve this horrible maths. And here we are. So basically, the purpose of this is what's really important to know about these materials is the size of the particles in them. So there's either a solid phase or perhaps in an emulsion we might have a fat in a liquid. And the stability or the performance of that material really depends on the size of these particles. So we can make the ultrasound measurements, but we need to then be able to work backwards to get the size of the particles. And as it turns out, we can use this sort of maths, which is an equation that tells us what happens when sound comes in and it hits one of these particles. And we solve this equation for this parameter here. Now, I've taken an excerpt from a paper Richard wrote. However, the numerical stability of the ECAH model depends on how the computations are done, and they can frequently prove to be unworkable. Yes, and I lost a lot of sleep on the fact that the computations can blow up. So I certainly learned a lot with regards to that. And this is the sort of classic work from Richard's lab where we have some experimental measurements. So you measure the ultrasound attenuation, you measure the speed, and solving that maths, we can work back to find out what size of the particles. So if we had an emulsion, we've got the oil, and if we've got a slurry or a suspension, we've got the glass. And we did a lot of work there, and however, I reached the point that I no longer wanted to work on the problem of the non-destructive evaluation of concentrated slurry. Sorry, Richard. And around that time, I heard about a healthcare revolution, something that was happening where we could cure conditions that were previously incurable. So I thought, I want to have a bit of that. So what was happening was this area of tissue engineering was developing, and people were having this engineered skin. So that was treating burn victims or people who had ulcers, for example, diabetic ulcers, which just didn't heal. We were looking at things such as a cartilage bone block to improve some problems with orthopedic disorders. A windpipe transplant that actually happened a number of years ago. We've got bone with marrow, so quite functional structures, and an engineered cornea. So this was like really exciting, and I'm thinking, as a physicist, how do I get involved in this? And at the time, there were two people, David Williams and Richard Archer, and they were both engineers, certainly David still is. And they had the foresight to know that although these tissue-engineered treatments were being proven in the lab, there was a big step to move it towards actual therapy. So you had to scale things up. You had to measure things about these tissues. Were they the correct properties? Were the cells alive, and so forth? And so David, he is quoted as saying, "With our engineering ability, we can be world-class in delivering regenerative therapies." And David really, I think, kick-started a lot of this. And Richard noting that of the scale of cells that we might need, 15 million cells to be grown. And so both David and Richard really played a big part. I know my career in the area of regenerative medicine and tissue engineering. And David is still very active, and unfortunately Richard passed away suddenly last year, so it was a real loss to the industry. But I was able to work with them on another postdoc position, and what that involved was sensing and control of these tissue-engineered products. So one of the products that were being made was a polymer that was used to plug the hole, really, where there was non-union in fractures. So if you had a bone with a very bad fracture that wasn't going to join up on its own, you could put the idea was you could put these scaffolds in. However, as you can see from the image up here, the variability of the properties of the scaffold is quite high depending on how you process it. So this was the task for us, is that in here, this is a high-pressure chamber, and this is where these materials were made. And so we rigged up some imaging systems so we could study what happens over time. And then we could quantify that and find out if we had a certain venting rate, so they put pressure in and then they let it out, and they've had carbon dioxide dissolved in this. So it's really like opening a bottle of fizzy drink and the bubbles coming out. So it's depending on how fast you open that bottle, depended on the size of the pores. So the quicker you did it, the bigger the pores, and there was some optimum parameter there. There was also interest in then looking at these scaffolds once you'd made them. So, and there was a big debate on how do you do this. And a parameter that was of particular importance to people was porosity. So how many holes, how well can cells get in, and how well can biological nutrients get in? But the issue is porosity. You can have one big hole or you can have lots of little holes. So it's not a very good metric. And moreover, people were using different methods. So we've got, that's an electron microscope image, we've got X-ray CT, that's MRI, and this is an optical method. So what we were tasked of doing is working out what's the best way to characterize these scaffolds to get metrics out that we could say this is a quality scaffold. So not only saying what the pores are, but perhaps saying how many pores there are and so forth. And then we went on to do some mathematical modeling so we could predict these parameters. So not very glamorous, but it was a way into this field of regenerative medicine. The other thing we got involved in was this is so Richard Archer set up a company called the Automation Partnership, and this basically is a robot in here, and in this area there are hundreds of flasks with cells growing in them, and so they wanted to scale up the culture of these cells. So if we need 15 billion or million cells, then we need to be able to make them in a way where we're not reliant on individuals. But they wanted to see what happened what happened in the process. So we made a mock-up flask, and we had we could measure the vibration, we could measure the temperature, the humidity, and the oxygen and the CO2. So we could track this process and see how it compared to if a human was doing it. Now, at the same time, in that lab, the cells that they were looking at were from a company called Reneuron, and they're a company down in Guildford. And their main product is a stem cell treatment for stroke therapy. So this has been in quite advanced clinical trials in Scotland. So was fortunate enough to work with them. And this is at the point where I realized that the power, again, coming back to the biological imaging. So these are some images that I took on behalf of a neuron. And the point here is that by having this image and insight into the biological world, we can start to understand the quality of these stem cells, how they behave, and how they can be best optimized for therapeutic purpose. So this was really a shift now to what is called cell therapy, where cells are used to help the body cure itself. And it really has moved from a situation where there's hope to it being a reality. And if we look at the global healthcare market per disease, there's tremendous scope here. So we were looking at cornea repairs before, so blindness, cardiovascular disease. There's tremendous potential with spinal cord injuries, Parkinson's, Alzheimer's, these sorts of conditions where we currently consider there's no cure for. Then there is this potential offered by cell therapy. And there's certainly investment going into this, and we can perhaps gauge the success of this field by the number of clinical trials that are starting. So that's starting to increase. So we're moving from the biological lab towards actual translation to the clinic. So what people normally do is they take images, these really pretty, colourful images of cells. And that's really great because it tells us what sorts of proteins the cells are putting out and a lot of functional information. But to do this, we have to put something into the cells. So we have to put some kind of chemical or some biological molecule that's not normally there. And sometimes we even have to kill the cells. So if we do that, we can't then put it in the patient. So there was a real need for us to look at some ways where we can study these cells, characterize them, and then have them still be viable for use in the body. So that's where we start making pictures. And in this case, I wanted to talk about some light microscopy techniques that were used. So microscopes, here we are at one I made the other day in the lab. So we've got here a syringe with a solution of diluted blood, some blood cells there we can see in the blobs there, and then we can see we've got a laser pointer and a syringe, and the droplet is acting as if we've got a lens. So if we use a laser which has a nice light source, we can see that the light from this laser will interact with those objects, the blood cells, and give us an image. But that's pretty simple. So a better microscope, we can make a better microscope, we can put some lenses in. And here I put a piece of red onion, and if we can have a look at what we get, I'm not going to win prizes in cinematography, but we can see that with the addition of some lenses and a very simple setup that we can start to get some images which give us actual structure. So if we consider what we can get from such a simple microscope, what can we get from a microscope like this, which we built in the lab? So the beauty of this microscope, well, there are many useful features here. One being that actually on top of the microscope stage is an incubator, so we can keep cells alive, so we can watch the cells as they're growing. We can also, we've got different arrangements of lenses and illumination, and so this means we can get images of multiple modes. So that means we can look at the way that light interacts with the cells and make different types of images. So say, for example, does the light go in and how much does it attenuate the signal? Or when the light goes in, how much does it shift the light beam relative to other parts of the beam? And by building up these different numbers of images, we can in fact get a really good insight into the biological system without having to add in these chemical dyes. Now, one modality which proved to be really useful was this approach where we harnessed total internal reflection of light. So you may be familiar with like optical fibers. So optical fibers send light down, and the light keeps bouncing in and stays in the fiber. And that's because when it hits the edge of the fiber, it hits it at a certain angle called the critical angle. Now, the angle that we hit it at depends on the speed at which light travels through that material. So if we enter the light in at a shallow angle, we find that we get refraction and the light will go through. So it would leave our optical fiber in that case. If we hit it just at the right angle, though, we get this total internal reflection, and in the case of the fiber, it would stay in. Now, what's interesting is that this angle here is very sensitive to the material above it. So if we had, for example, the culture fluids, the fluid that the cells grow in, and we had a cell, we find that just by having the material of the cell near that substrate, that we can get this frustrated internal reflection, which means the light carries on. So what that means is that cells near the glass substrate appear dark. And we can probe the membrane of the cell, which is a very important part of the cell as to how it functions. And just to show that, so if we hit at this shallow angle here, we don't get any reflected light. And when we hit this magic angle, we get reflected light. And we can see that if we have this culture media that the cells grow in compared to the material of the cell. So if we hit at some intermediate angle, we'll see where we just have the media, we'll get a big reflection, and where we have the cell, we won't. And that forms this contrast. So this is the optical path of the microscope. And we've got a number of different arms coming through. We've got a few different fields of view for imaging. And it's this area which helps us select the angle that we want to illuminate at. And then from above, we illuminate, and it's this device here called a spatial light modulator, which enables us to switch between different modes of imaging. So it's quite a convenient setup. So, contrasting images. So the different images give us different information. And here we've got an image of this is a mouse neural cell. And this is called bright field transmission. So it's the light has gone through, and we're really looking at how much the light has been attenuated. And typically with cells, it's not much. So the contrast in this image is quite poor. But if we use that total internal reflection approach, we get much, much more contrast. And it shows you how sensitive this refractive index change is to the reflection or non-reflection of the light. And we can in fact see in tremendous detail the filaments that are coming off these neural cells. Now, I said multi-mode, so not always one mode's the best. So here we're looking at the bright field transmission, and this is a useful way to count how many cells there are. So we can see quite clearly there's a cell, there's another cell, and so forth. But if we go to the total internal reflection image, because I said we're just looking at the membrane, so it's like we're looking at the footprint of the cell. This is great to look at the membrane, but if we want to count cells, rubbish. So by combining these multiple modes, we get a real powerful set of information. And we can take some time lapse, and because we've used that spatial light modulator, we can get the 3D morphology as well. So we've sat a cell down in the dish, and this is over a 2.5 hour period. We can see that the cell's exploring its neighborhood, if you like, and we can see how the footprint of the cell is moving about, but we can also see how the thickness of the cell is changing. So having this sort of information is really helpful for those developing these stem cell therapies, so they can optimize the way in which they grow the cells, and they can understand how they function better. And this just reiterates we've got this complementary information. So here we've got a co-culture of, we've got some neural cells, so they're from the brain cells, but we've also got another type of cell, a brain cell called a glial, so rather an astrocyte. And they're both in this image, but it's rather difficult to see the astrocyte in this view. Again, we're seeing the neurons. These are these sort of cells with large bodies and long projections. And then we look at the total internal reflection image, and we see there's that astrocyte. So the astrocyte was hanging around right at the bottom on the surface. So it shows that we can get information about the neurons, information about the astrocytes, but we need both, because if we just had this one, we wouldn't know we had neurons and vice versa. And then we can overlay them, and we can start to build up a nice pattern of how these cells are actually growing in culture. So it's one thing to make pretty pictures, but we also want to give some quantitative information. So we want to inform the people who are making these cells and say, look, at certain phases in the culture process, we know the cell is in a certain state. So we took those images and we took some measurements from the images, and we worked out a strategy that would enable us to say, if they fall in this area, then they're stem cells. Up here, we know we've got neural cells and a mixture of neural cells and astrocytes, and up here we've got a distinct area of neurons, and that gave us a strategy. So we can say, by having these measurements, we can sort of say, if this is true, then we've got an astrocyte, and we can go through a flow chart, which we can give the people who are making these stem cell therapies to help them be able to monitor the process of their cultures. But we come back to this engineered tissue, and this is still a really important part in medicine and in part of the therapies. But you may notice, apart from the cornea, they're pretty opaque. So if we want to think about how we might look at this with microscopes, we need to take a different view. So the problem is that these tissues scatter light, okay? And when we scatter light, we lose resolution, we lose the information as to where objects are. So one approach that has been used is something called optical clearing. And basically, they take the tissue and they put certain chemicals on it, which basically strips out this part of the tissue that's scattering, and then they can get an image where they can see the cells in 3 dimensions. So they've got the tissue where the cells are, and the way that they want to be able to see that with the microscope is to get rid of this scattering, and they do that chemically. But again, we're all about non-invasive. We don't want to put chemicals on the system. So what do? Well, we better make a microscope that can handle that sort of situation. And what we use is we use a microscope which has a cylindrical lens rather than, I guess, your traditional circular lenses. And a cylindrical lens, when we send the light in, it makes a sheet of light. Okay, so what that means is that if we have our sample here, we can move our sample up and down through this sheet of light, and we can take a picture at each position. So it's almost like we're taking like a CT scan or an MRI, so we're getting a slice of each image. And then we can know where, when we're at a certain axial or certain height, what is in the image. So this is an example here of if we don't do that, and we've got some polystyrene beads. So this is actually the same sample. So if we just send light straight through, we can't work out that there's more than one bead or what on earth the structure is. But if we image with this sheet of light, then we get this axial information. So that's the use of this selective plane microscopy. So we wanted to basically show that we could achieve what people have been doing with optical clearing, but without having to optically clear. So this is a gel which has got cells. So these are lung cancer cells in them growing here. And you can see with the traditional image, so we've cultured this system over a week, we can see, well, nothing very useful whatsoever. But if we use our selective plane microscope, we can build up a 3D map, and we can find out where these cells are and the density of them. So that, again, that's really important in terms of growing the tissue, because you want a certain amount of cells for a functional piece of tissue. And if we go back here, it's basically we've managed to achieve this without having to go through those steps. So it's quite a useful technique. And so this is Mike Somack. He's a professor at Nottingham I worked with, and he's really been the driving force behind that and my work in the microscopy. And I'm pleased to say that through the Institute of Liberal Arts and Sciences that he'll be visiting Keele in the summer. So I hope to have some very useful exchange over that period between his lab in Hong Kong and our lab here in Keele. So, we still want to make pictures, but this time we've exhausted our abilities of light. So we need to do something else. And so what we find is if we add sound to light, we can see even deeper. So if we think about biomedical imaging, so when we say that, we're talking about human tissue, sort of people-sized structures. And you can see if you put a light behind your hand, it just goes everywhere. And so that's because of this problem of scattering. Nonetheless, biomedical imaging with optics has an important place. And here we can see that it's able to map the vascular system, so look at the blood flow in superficial structures. And we can see here we've got a photograph of a skin cancer cell, and this is an optical method called optical coherence tomography, and we can see by scanning that we can identify this tumour. So biomedical optics is certainly an important field on its own. However, we're limited here by the order of a few millimetres. Now, for something to be clinically useful, so if we're thinking about clinical use, and if we wanted to image some breast tissue, for example, we're looking at needing about 5 centimetres or something. So with optics alone, we're not going to get there. So we bring in the sound. So why do we use sound? Well, the problem that you saw with the light was it scattered. Now, the amount in which it scatters depends on the size of what's scattering and the wavelength of the light, if you like. So as it turns out, sound has got a much, much longer wavelength than light. So it means it scatters less. So what we do here is we send in sound and we focus it. So we can focus to a much tighter spot with sound. Then we send the light in. And what happens is when the light interacts with the sound, it changes its intensity. So it interferes and interacts. And so we can see if we measure the intensity here, we get this slight wobble. And that slight wobble is due to the ultrasound. And if we have a look at it in the frequency domain, we can see this is the optical frequency, and we can see that there's these other components which shift the optical frequency by the ultrasound. Now, why that's useful is if we just lock on to these sidebands, then we know that that light has gone through this position, this focal position. So we can pin it down. So rather than thinking we've measured the light here, we don't know where it's been, we know at least it's gone from this point through this focus and to this point. So we can improve our spatial resolution. So as an example here, we've got some absorbing targets, so some black marker pen on a transparency, and we filled it up with a gel. So this is to mimic tissue. So as you can see, once we've put this gel in, you can't see these targets anymore. And that's just a close-up, but this is a line scan. So imagine we were scanning our sensor across here. So without ultrasound, we just end up with one single blob. We can't resolve there are two marks. When we turn on the ultrasound, we can improve it and we can resolve those two features. And if we use an even higher frequency of ultrasound, we can improve that spatial resolution even further. So that's all well and good, but that's just a piece of gel. So being physicists, we went out and got some chicken breast rather than culture it ourselves. So again, the same transparency inserted inside this piece of chicken. And you can see with ultrasound off, there's a general blob suggesting there might be something there. But when we put the ultrasound on, we can distinctly resolve these two areas. And so this really has sort of, I guess, taken biomedical optics into a deeper area of imaging and making it, I guess, more viable or opening it up to further applications. But there is still an interest in this fluorescence imaging. So, earlier I was saying about cells, we don't want to put chemicals on, we don't want to damage them. But there is a role for these fluorescent markers, and the reason is they're very specific. They can tell us what sorts of proteins the cells might be putting out, and we can link that to a disease. So certain cancers will give out certain proteins. So we can quite specifically say what the cells might be. And this is particularly useful in preclinical models. So we're not studies that are going into humans, but these preclinical models that can help us refine the treatments. But the work that I've just shown you used a laser. And the situation with the laser is it has a property where as the wavefronts go along, each wavefront has a constant relationship with the wavefront that went before it. And that property is something called coherence. So lasers are very coherent. And that means that they mix or they interfere very well. So when we put the sound in, that these lasers will interfere well and we'll get that modulation. The thing about fluorescence is it's incoherent. So the relationship between one wavefront and the next is random. And what that means is that the modulation is very weak. So what can we do to overcome that? Well, we had a look at a way of using this ultrasound modulation in a different approach. So these are some gas bubbles. Okay, so we've got like a lipid, so you've got a fatty outside and you've got a gas inside. And here we've got some diagrams where ultrasound has been applied. So ultrasound can squeeze down the bubble, make it bigger, and squeeze it down again. So the ultrasound can do this to the bubble. Now, if we can put these fluorophores, these fluorescent probes, in the shell of the bubble, we can then change the proximity of these fluorophores to one another. And it turns out that when you get lots of fluorophores very close to each other, they turn off. So by applying the ultrasound, this is some simulations here. So here we go. We've got some mathematical simulations here. So we've got the radius of the bubble changing with the pressure of the ultrasound, and we've got the photons or the amount of light also changing. So we can basically turn the light on and off. And we can get our signal here where you can see this is this sideband we locked onto, and you can see we've got this frequency. But to be honest, that's a pretty noisy, rubbish image. And there is a problem also with these gas bubbles is they're not stable. They disappear in the body after maybe 10 or so minutes. And because of their micron size, they stay in the blood vessels. So can we do better? Well, what we did then is we used liposomes. So liposomes are like a cell. They're like a human cell, but with nothing in them. So they've got a bilayer of fats around them, and they've got a liquid inside. And we can make these very small. We can make them on the nanoscale. So in a very similar way, we did two approaches. We actually put these special fluorophore labels on the lipids, and then made the liposome containing these fluorophores in. And then another approach was we integrated these fluorophores in the lipid itself. And when we put the ultrasound on, it changes the proximity of these molecules. And this proximity, as I said, is highly related to whether the light is on or off. And so we can see here that when we turn the ultrasound on, we get the light on, and when we turn it off, we get the light off. And we can do this in a cyclic manner, and it means that we can lock on to this ultrasound and we can improve the spatial resolution. So we move on to the last chapter really of the work which relates to the current project that I'm working on. This is this project that's funded by the European Research Council. And so making pictures, quantum diamond microscope. Right, this took me quite some time to work out how I may explain it, and I, in the process, thought I don't understand it anymore, but here we'll give it a go. So basically, what we're trying to do is we're trying to look at the transmembrane protein. So all our cells in our body have this plasma membrane, and it's the outside barrier of the cell, and it really protects us. It's sort of the gatekeeper, if you like, of what goes in the cell and what comes out of the cell. And in this membrane, we've got a lot of proteins that are sort of represented here by these different colors. These proteins play a really key part in how the cells function, and in fact related to their dysfunction. So we have some transmembrane proteins called ion channels, and they, with a voltage, can open and close, and they're related to cells like muscle cells and heart cells, so you get this electrical pulse. We have other proteins which are receptors to things, to other biological molecules in the body. And one of the sort of leading scientists, Lloyd Hollenberg from the University of Melbourne, in this field of quantum sensing, I guess has really clearly put it that the ability to image the atomic structure of molecules in their native cellular environments is vital to both understanding disease's origin and finding its cure. So the key here is to have a look at it in its native structure, and currently there are only about 30 out of 800 known human membrane proteins defined, and the reason is the current methods to characterize them involve we have to take them out of the membrane, we have to try and form them into a crystal, or use other techniques where we disrupt the protein from the membrane. And the function of these proteins really depends a lot on how they're folded. And it's like a coiled spring in a way. If we take them out of the membrane, then they will sort of uncoil, and we won't fully understand their structure and therefore how it relates to function. And the reason this is important is really for curing disease and drug discovery. So about 2/3 of all drugs currently in development target these transmembrane proteins. So they could, if we understand these proteins more, we can really improve these drug targets. So now we come on to the quantum sensor. So imagine we have an electron. So atoms, we've got atoms have got electrons in them. And electrons can act like a tiny bar magnet. Okay, so they've got this quality called spin, which means they sort of spin around their axis, and that makes them act like a tiny bar magnet. And what these spins can be is they can be the up or they can be down. And that's what we call a classical system. So these electrons can have an up spin or a down spin. But in a quantum sensor, they can have both spins at the same time. So it's called a quantum superposition. And when we measure it, when we measure the spin, we can find that there's certain chances of us finding the spin in the up state or the down state. So what's the point of this? Well, these spins only stay in this quantum superposition for a certain amount of time, a short amount of time called the coherence time. And we can knock this state out by changes in the external environment. And that's the real key sensitivity of these quantum sensors, is that they are very sensitive to their external environment. So if we take these sensors and we slightly change the environment, then they will respond significantly. So that's the power of the quantum side of the sensor. And it turns out that there are certain defects in diamond which contain these, can be considered as these quantum sensors. And in fact, the one way of looking at it is called a nitrogen vacancy. So in the atom, in the lattice structure of a diamond, we've got carbon atoms. And if we knock out two of those and put a nitrogen one in, and we leave the other area empty, so it's a vacancy, we find that we can get an electron spin inside this diamond that can act like a quantum sensor. Right, so we've got that. So we've got a diamond and we've got a quantum sensor in it. Right, what do we do next? And why is that useful? Well, it means we can do something called nanoscale MRI. So most of us are familiar with an MRI machine. Person goes in and you get this image, and you get spatial information of the order of millimeters or so. And one of the reasons for that is that to detect the magnetic fields that are associated with producing that image, they use these big coils. So there's these big coils in here which detect the magnetic field. But the thing with the quantum sensor, so this is this electron spin, this is the nitrogen I was talking about, and this is this vacancy. And so, as I said, this spin can have up and down, or this superposition of spins. The quantum sensor can detect very small magnetic fields. In fact, it can detect, its sensitivity is about 1 million times more than that of the best MRI machine. So that means we can start to think about actually sensing these biomolecules associated with the cell membrane. And a very important point for us in terms of microscopy is that there's an optical readout. So with these particular diamonds, what we can do is we can shine green light on them, a certain color of green, and we can change the spin state. So we can put this electron spin into, for example, the down state. Then we can apply some microwaves, and we are able to put it into this superposition, so we can put it in this up and down state. And we find that there's a certain amount of energy needed to flip this spin from the down to the up state. And that amount of energy is where the resonance comes in, and in any electron spin resonance or nuclear magnetic spin resonance. So what we can do is we can scan the frequency, and here we're looking at the light that comes out. And we find when we hit the right microwave frequency, we get a big drop-off in the light. So it means that by reading out and using the microscope to measure the light that is coming out of these diamonds, we can work out what sorts of molecules we had. Because when we have, for example, we have certain nuclei, we might place one of these nanodiamonds near a protein, and the protein will have an influence on the magnetic field that the diamond senses. So if we had, for example, a protein nearby this diamond, we would see a change in this graph. So this is how we can start to use these diamonds to sense. So as I said, the external magnetic fields from the nearby electrons or protons change that fluorescent readout. And we can image that with a microscope. So that's really the basic science behind this 5-year grant that we've got from the European Research Council. And it's all about imaging transmembrane proteins. And as I mentioned before, they're really essential for life, and essential that they're characterized in the natural state. And this is just a schematic idea of what we plan to do. So this is a cartoon of the cell membrane here. And this is a cartoon here of these ion channels, of these proteins. And if we can use some biochemistry and get these diamonds close enough to these proteins, then we can sense the magnetic fields that these proteins are putting out. And so by making measurements of the amounts of red light we get from these diamonds when we shine green light, we can start to work out the structure of these proteins. We can also look at seeing when these proteins function. So when these ion channels open, you get a flow of ions, and that makes electric current, and related to a moving electric charge is a magnetic field. So the mere opening of an ion channel will cause an external magnetic field, which we hope to be able to sense with the nanodiamond. So it's a tremendous privilege to be able to have this funding, and certainly if we can go even partway towards realising this goal, it will be a very good achievement. In my career, I've found that it's one thing to secure the funding, but there's another thing to find people to work with. And I'm very fortunate in this instance to have Valentine, Josh and Simon working with me on this project, and I think we formed quite a good team, and I'm quite optimistic we'll be able to make some real progress. So that leads me to the end, really, of my presentation. And I wouldn't be standing here without, the support from many people. And I hope I remembered everybody. And particular thanks to my partner Di, because without her, I wouldn't be here and wouldn't be in this position. So thank you very much for that. And I must also remember to thank the funding bodies, because again, without them, none of this would be possible. So thank you very much for your attention.